Howdy Bike 410 students. All right, this slide is from the Posey Notes, which I said before will pop up on your Chapter 12 module in the next week. And what we're going over in this Posey slide is the classification of enzymes by the type of reaction they catalyze. So oxidoreductases catalyze oxidation-reduction reactions, so the movement of electrons. Number two, transferases catalyze the transfer of functional groups. So for example, a phosphate from the gamma phosphate of ATP might be transferred onto a sugar or onto a protein's serine, threonine, or tyrosine, and that would be catalyzed by a transferase. Hydrolases you've seen before are toolbox proteases, chymotrypsin and trypsin, for example, catalyze the addition of water across the peptide bond, and that is a hydrolysis reaction. So that's the type of reaction that hydrolases catalyze. And lyases, which we won't have an example of, are those enzymes that catalyze the elimination of a functional group and the formation of a double bond in place of that other bond that's lost. The isomerases are a class of enzymes that catalyze isomerization reactions. So, for example, if you were moving from glucose to galactose, that would have to be catalyzed by an isomerase because you have the same number of atoms, the same number of bonds, the same composition of atoms, but they're in a different configuration. And we'll see an example of an isomerase in the required video for this module. And finally, class 6. Ligases are enzymes that catalyze the formation of a bond. And so you might think of when you need to move a gene from one organism into bacteria for recombinant DNA technology to express a protein of interest. We do this in the lab all the time, and it requires energy. So in order to make the phosphodiester bond between the DNA fragment that you cut out with your restriction enzymes and the DNA that you cut with restriction enzymes to accept that fragment, you would need a ligase to speed up the formation of the phosphodiester bonds to reseal those double-stranded DNA parts. And that costs energy. So one of the characteristics of ligases is the requirement for ATP hydrolysis. So make your slides, your flashcards with each of these different classes, and anything that is special about that class of enzymes should go on the back. All right, so now we're looking at the actual substrate in a schematic form here in yellow. And you can see that this substrate up here has different functional groups. So it has an oxygen. This O right here is an oxygen. So this has a partial negative charge. And then there are hydrophobic groups shown in brown here and here. And so they will be favorable against other hydrophobic groups. And then you have a good proton donor right here with a permanent partial positive charge. And then you have a formal negative charge in red. And so that means that when your substrate binds, as it is bound down here, this is your bound substrate, it's going to partner up with parts of the enzyme active site that are complementary to it. And that gives the enzyme specificity. Okay, so when we say specificity, we're talking about the enzyme's ability to discriminate between one substrate and another. And so this enzyme will only bind to this substrate because it has the right shape complementarity, the right polar complementarity with all the hydrophobics facing each other like this and this and this, the right hydrogen bond donor and acceptor complementarity, and charge complementarity. So all of those types of complementarity that we've talked about all semester are going to come into play at the active site of an enzyme when it binds its substrate with such specificity. How can an enzyme be so specific? That question is addressed in this slide, and we're looking at hexokinase. This is a transferase molecule, the enzyme hexokinase. It has two domains, the dark blue and the light blue domains, and the active site cleft is right in between the domains. And it's big enough for all of these different substrate candidates. So if these are all substrate possibilities, glucose, glycerol, and water could all fit in there, right? But only glucose actually has the right shape and complementarity as the active site. And in fact, only glucose will trigger the conformational change where the domains actually come closer together and exclude anything else from binding. So that once glucose is bound, there's not even any accessibility to water. So water can no longer bind here. That's what is meant by solvent inaccessible active site lining. Okay, so yes, these other molecules are smaller than glucose, but they don't bind with the same specificity, so they won't trigger the conformational change required for hexokinase to activate its groups and catalyze its reaction. We call that induced fit, that ability of enzymes to move once their substrate is bound in order to engage all of their groups for that good, precise fit, so that they are specific for one and only one substrate. Some enzymes are so specific that there is stereospecificity. So you can have different shaped and, of course, differently charged and different van der Waals surfaces, etc., that actually only fit in one way. So this molecule will only bind in this way, not in any other orientation. So not only would this enzyme only recognize its substrate, but it only recognizes its substrate in one orientation. And we'll see an example of a very stereospecific enzyme when we get to the isomerase video example. This is the isomerase video example. It's the PDB video that's already up in the Chapter 11 module on eCampus, and this one is actually required viewing. I will probably be taking a question from this video for Quiz 10 and or exam, the final exam. So make sure that you watch this video. Aconitase is an isomerase, and it acts by converting citrate into isocitrate. And the only difference is that one of the H's so if we get rid of this too, one of the H's on carbon 4 is going to switch places with this hydroxyl group on carbon 3, the green one. And you can see that over here in isocitrate where the H and the OH have switched places. And how that actually works is what this video, the PDB video, is about. And these PDB 101 is an excellent source to know about if you ever go on, let's say, to medical school and you're taking a class on drug design, then you'll be very happy to know the PDB 101 website. So go ahead and look at that. Now, other enzymes are not quite as stereospecific. So, for example, our teach is going to be about your toolbox enzyme chymotrypsin. And chymotrypsin is not an isomerase, it's a hydrolase. It adds water across the amide bond in a peptide, but it also will catalyze the addition of the elements of water across an ester linkage. So it's going to actually recognize the R group. You know this, you remember this. And it will, once it's recognized a large bulky hydrophobic group at position R, it will catalyze the addition of water across the peptide bond that follows that side chain, right? And so it works on an ester as well as it does on an amide. And that fact was actually discovered early on in the analysis of serine proteases. And so we'll be talking about the ability of chymotrypsin to cleave an ester when we get to the teach. And that's where this video ends. And the next slide is where the next video begins.